After William Morton Wheeler's departure from UT in 1903, it only took 5 years for a new direction to take shape, through John Thomas Patterson. People that knew Patterson affectionately referred to him as Dr. Pat, and always used interesting language to describe him. Short and plump, he looked and behaved like a bulldog. Sort of pear-shaped, but not a couch potato. And perhaps somewhat more eloquently, sandy-haired and short of stature, he had a ready wit, a love of repartee, and the ebullient temperament we had traditionally associate with the Irish people. That was from Theophilus Painter in a 1965 memoir about Patterson. Patterson's energy served him well. He started at UT as an instructor in 1908, and in just 3 years was promoted to zoology chair. In many accounts, Patterson was responsible for converting the zoology department at UT from obscurity into one of world renown. This was in no small part due to Patterson's push for research on the genetics, evolution, and diversity of the fruit fly, Drosophila melanogaster. Patterson was trained as an embryologist, but during his early years at UT, he became increasingly interested in the field of genetics and wanted to put Texas on the map in this area. He was familiar with the groundbreaking work of evolutionary biologist and geneticist Thomas Hunt Morgan and his students in the famed fly room at Columbia University. One of Morgan's students in particular had Patterson's eye, Herman Muller. Patterson was impressed with him and set his sights on convincing the young Muller to come to Texas. This would require arranging some perks for Muller, like fully funding his research supply needs, which was not a common thing back then, providing an assistant and eventually a larger fly room, and a special smaller constant temperature room to keep the flies cool during the brutal Texas summers. But why were fruit flies the focus of genetic study at this time? Drosophila melanogaster is a good species with which to study genetics. It has a short, simple reproduction cycle, allowing several generations to be observed in just a few months. With 4 pairs of chromosomes, it has relatively simple genetics. It has a compact genome that is easy to manipulate genetically. These flies are also easy to keep. They are small, but not so small to see without a microscope, which allows researchers to keep millions of them in the lab at any one time. Muller would come to UT in 1920 and leave in 1932. Not unlike the rest of his life, these 12 years were both professionally and personally intense for him. However, much of the research that led him to the Nobel Prize in Physiology and Medicine in 1946 was done at UT in the 1920s. As Clarence Paul Oliver, a research assistant of Muller's at the time, stated, Muller reported results in 1927 which caused his reputation and the department's recognition to take a big step forward. He showed that mutations of genic material can be induced in Drosophila by exposing them to X-rays. Muller did this by devising a mating scheme that simplifies tests for sex-linked lethals, or any visible sex-linked mutant. Along with later reports on the effects of radiation on genes and chromosomes, this discovery reset the direction of research programs in the department. Only a few years later, after Muller's departure in 1932, Patterson would tire of Drosophila genetics. Activity continued in the fly room, in the biological laboratories building on campus, but largely through the work of postdocs and graduate students who would handle the tedious work of dealing with countless etherized flies under a microscope. However, it only took a few years for Patterson to return, rejuvenated by exploring speciation in Drosophila. He and his graduate students began ambitious collections of other Drosophila species, shifting the fly room from its focus on melanogaster to the most extensive survey of Drosophila species ever attempted at the time. Another important figure in this group was Wilson Stone, who would emerge as the intellectual force behind many studies and a leader in the zoology department. In 1951, the genetics group moved from the biological laboratories building on campus into the new experimental science building, which was demolished in 2008. The Experimental Science Building, or ESB, provided each researcher a separate lab, and the dynamics of the old fly room as a singular location did not live on. One of Patterson's graduate students, James Crow, had suggested that Patterson was negatively affected by this move, and that he probably missed the interactivity of the old fly room. His research zeal quickly diminished, Crow would state. Patterson would die at the age of 82 in 1960. Well before his death, however, other insects became research subjects at UT. Part 3 of this video series looks at mosquitoes, insects that we in Austin know quite intimately.